Okay, let's continue to look at the differences, okay, between meiosis 1 and meiosis 2. Okay, in prophase 1 and prophase 2. Okay, chromosomes with double helix, chromosome with double helix. Okay, all these are the same. Homologous chromosomes go through synapsis process. Okay, remember the process is known as synapsis, the pairing of the homologous chromosome, and they are becoming bivalent or tetrad. Okay, the four sister chromatids, and the number of chromosomes is 2n. Okay, diploid. It is still diploid, just the first phase, and crossover happens here. Very crucial process that happens in prophase 1, but in meiosis 2 there are no homologous chromosomes. Why? Because the homolog chromosomes already separated, okay, in anaphase 1 just now. Okay, and the number of chromosomes now, they are haploid, and no crossing over happens. Okay, next, in metaphase 1, homologous chromosomes are now arranged at the equatorial plate. Okay, equatorial plane. Okay, so metaphase plate, last time when the old syllabus, we are using these terms. So we just follow the textbook, the new syllabus, they are using the term equatorial plane. So centromeres are still attached. Okay, centromeres not separated, right? And the chromatids are remain together. This one is referring to the sisters chromatids. But in metaphase 2, chromosomes are aligned on the equatorial plane. Chromosomes no longer homologous chromosomes. Okay, and the centromere here separated, and the chromosomes are sorry, the sister chromatids separated. Okay, in anaphase 1, homologous chromosomes separated towards the pole, sister chromatids remain together. But in anaphase 2, identical chromatids now they begin to separate because of the separation of the centromere just now, and they are moving to the opposite pole of the cell. And in telophase 1, nuclear membrane reform. Okay, same goes to telophase 2. Okay, and now the chromosomes that are formed is haploid, double helix, but here it is haploid with single helix. Okay, let's compare now between meiosis and also mitosis. Compare. Okay, so there's a video clip for you to watch. Okay, mitosis versus meiosis. So, place of occurrence. For mitosis, it happens in all the somatic cells. What are somatic cells? They are the non-reproductive cells. Okay, whereas for meiosis, gonadic cells, referring to the reproductive cells. Okay, the role or the significance or the need. This one is to produce cells for growth and tissue repair. Okay, so that means you want to produce identical or the same cells. That's why the number of chromosomes is the same. Right, but here for meiosis, what is the need? You want to produce gametes, and you want the gametes to have genetic variation. Okay, the number of divisions is only one stage of mitosis, but in meiosis, okay, there are two stages, two divisions. Synapsis of homologous chromosomes, okay, it is insignificant in mitosis, but in meiosis, it happens in prophase 1. Okay, very important phase, and it forms tetrads. Crossing over is the name of the process. Chiasma is the name of the point where they cross over. In mitosis, this one is tidak berkenaan, but in meiosis, it happens in prophase 1 of meiosis 1. Okay, the number of daughter cells formed at the end of division for mitosis is 2. For meiosis, it is always 4. Okay, number of chromosomes in mitosis, diploid 2n, same number of chromosome, and the genetic content is same. This is really a photocopy. But for meiosis, okay, half the number of chromosomes of parent cell, and they are not the same, the genetic composition, because of crossing over just now. Okay, because the one of the need just now is to form to produce genetic variation. Yeah, this is the one. Okay, ploidy level. So that means ploidy. Ploidy here refers to haploid, diploid. So remain, this one is 2n, the daughter cells also 2n. This one parent cells, and it is further reduced. This is at the end of telophase 1, you get 2 daughter cells, haploid. At the end of telophase 2, you get 4 daughter cells and all haploids. Alright? Okay, so another picture for you to observe, okay, to study. Alright, so let's look at this self-assessment.